Good afternoon, good morning. Welcome. My name is Gina Olson with the Muscular Dystrophy Association's Healthcare Partnerships team. Thank you for joining us for MDA's Industry Update webinar, The Promise of Gene Therapy in LGMD 2I R9, an update on the AskBio Lion 101 study presented by AskBio. MDA's Industry Update webinars are programs which provide an opportunity for the community to receive information and updates impacting care and treatment directly from pharmaceutical, biotechnology, and medical equipment companies. These webinars are just part of MDA's educational resources. We encourage you to visit the Community Educations page on mda.org. You will find an abundance of resources, information, recorded webinars, and listings for upcoming educational events. Of course, if you have questions or need assistance, the MDA Resource Center is available to help you by email at resourcecenter@mdausa.org or by calling 1-833-ASK-MDA-1, or you can reach the Resource Center on mda.org. At the end of this webinar, you will receive a survey. Please take the time to complete the short survey so that your voice is heard and you can help us create more impactful programming for future events. Now, I'd like to turn it over to our speakers, Ina Shah from AskBio for today's webinar.
Good morning, good afternoon, everyone. So excited to be partnering with the MDA, as well as our patient advocacy organizations, as well as our healthcare providers to provide you this very important webinar on gene therapy and the promise in LGMD2I. Little bit about AskBio. I'm joined at AskBio by Ann Nguyen, who is our Vice President of Therapeutics, Kara Witkoff, Director of Patient Advocacy and External Engagement. AskBio is a Bayer cell and gene therapy platform company. We're considered leaders in next generation AAV gene therapy and a pioneer in almost every element of manufacturing as well as commercialization. We were founded about 40 years ago in 2001 by Jude Samilsky. And we have a clinical pipeline that covers a broad range of therapeutic areas, including central nervous system, muscle, respiratory, and heart disease. We have more than 500 employees throughout the world. And we've also worked with other therapeutic areas as well as companies in order to provide our technology and assets. Kara, if I can turn it over to you.
Thanks, Ina. Ask First is our collaborative program for advocacy groups, patients, and their families who may benefit from our gene therapy research as we strive to advance innovative therapies. We ask first how we can help patients and their families, and we welcome your voice to help us erase genetic disease. Guided by the voice of patients, Ask First is driven by our mission to improve patients' quality of life and to deliver genetic medicines to treat unmet medical needs. Now to our presenters. Kelly Brazo is the co-founder and CEO of the Cure LGMD 2I Foundation, established in 2011 after her daughter Samantha was diagnosed with limb-girdle muscular dystrophy type 2I R9. Collaborating with fellow advocacy groups and stakeholders, Kelly's passion is advocating, spreading awareness, and promoting research and drug development for this ultra-rare and progressive disease. Dr. Nicholas Johnson is the director of the Center for Inherited Muscle Research and vice chair of research in neurology at Virginia Commonwealth University. He received his undergraduate degree in molecular and cellular biology and psychology at the University of Arizona, as well as his medical degree. He completed his neurology residency and combined fellowship in neuromuscular medicine and experimental therapeutics at the University of Rochester. He also leads the GRASP LGMD Consortium, which focuses on developing clinical outcomes and biomarkers in the limb-girdle muscular dystrophies, and co-leads the Myotonic Dystrophy Clinical Research Network, which focuses on developing clinical outcomes and biomarkers. Both networks include sites across the US and globally. Next to Kelly.
All right. Thank you so much for having me. Again, my name is Kelly Brazo, and I have a daughter named Samantha, who's 15 years old and is living with LGMD 2I R9. And so a little bit about our story. When she was about 1.5 years old, we started having a lot of questions about her development. She was tripping and falling a lot. And when she did, she had trouble getting up off the floor. She was struggled at things like gymnastics and dance, which were her favorite activities. And she did then what we know now is called the Gower sign, and would actually grunt to get up off the floor. And so that sort of led us down the line of what's going on. Maybe she needs some physical therapy. So we saw pretty much every specialist, what felt like up and down the East Coast, lots of appointments. We found that she had elevated CK levels, and heard several times, well, if she were a boy, I'd swear she had Duchenne. So we thought we had muscular dystrophy ruled out. But lo and behold, we didn't. We ended up at the NIH with Dr. Bonneman, and he ordered a LGMD genetic testing panel and found that she did have LGMD 2I. And worry at the time was very discouraging. We were told, well, there's not much you can do. Keep her healthy, and you can try prednisone if you'd like. And that wasn't good enough for us. So we are thankful for what has happened as of the last couple of years. But anyway, so when she was little, before the effects of LGMD2I really took over, we kept her as normal as possible. We tried to get her involved in as many activities as she wanted. And someone gave me good advice. A friend of mine who's an occupational therapist said, don't be hypervigilant. Let her do what she can do. Let her enjoy all the activities she can while she can. So we really took advantage of that advice. She was a pretty good little softball player. My husband taught her to pull it to third so she could make it to first base just barely in time. But more recently, in the last couple of years, she wasn't able to do sports anymore, but she started horseback riding, which was really fun for her. And she took an electric scooter on family outings and vacations to try to keep up her energy. So everyday challenges for a sophomore in high school, things that she does have to worry about now are falling. And when she does fall, unfortunately, she's not able to get up on her own. She's gotten stuck in the elevator at school. She does miss out on a lot of social events, unfortunately, just things like Friday night football games. The bleachers are not kind to her. She doesn't participate in gym class at school. She doesn't play sports. She often rations her water intake because it is difficult for her to get to a typical bathroom. And, just in general has lost a lot of her independence. But I'd say the biggest challenge that we have faced is contractures. And about 2.5 years ago, she had her first surgery. She had a bilateral heel cord lengthening procedure and wore casts for about 4 weeks after that, and pretty much had to, relearn how to walk again. But thankfully, it was successful. She's able to keep her feet flat on the ground now, and she is much more willing to wear her night splints because she does not want to have that surgery again. So that was successful, I'd say. And 2 summers ago, she did have a massive spinal fusion surgery for her spinal difficulties. And that was tough. She was in the hospital about a month, and I'd say it was a very successful surgery as well. The thing that she lost from that is she's no longer able to hyperextend her back. So she's lost the ability to get up from a typical seated position because that was the way she was able to do it, was to really hyperextend her back and walk up her legs. And so she can't do that anymore. But otherwise, I'm going to let her tell you her story because she's 15 and she has her own opinion of how life is working for her. So I'll let her talk here for a second.
School's kind of like figured out now. I kind of had like a little routine that I do, but especially if I'm doing things with my friends, it's always like I like to go to like pick them up first so I can like kind of see like the outside of their house. I'm like, okay, she has like 5 steps but no railing, so like maybe there's like a back way, or like through the garage there's only like 1 step or something, and they're like I can use like the door to hold on to or whatever. But then it's also like, oh, like who's driving? Like that they have a little car, so I'm not going to be able to get out of that. But then it's also like, if there's all, like not everyone can help me. So I don't know who else is going to be there if anyone can help me. So it's just those kind of things. You always, like, you literally have to think about everything. Or if you're going to get food, you have to make sure there's tall chairs there. Or again, someone that can help me. Like, it's just all of those things that just like, have to go through your brain. Like you have to think about it. School's kind of like,
oops, sorry about that. So she did talk about school being figured out. So some of the things that we have, figured out as a strategy for school to be successful for her is she does have her own personal elevator key. She has a walking buddy to carry her books for her between classes. She leaves class a few minutes early so that the hallways aren't crowded and doesn't get bumped into. She also has duplicate textbooks for home and for school. She has 2 lockers, one on each floor, so it's easier to access that when needed. And she has bar stool height chairs and tall desks in all of her classrooms, as well as in the cafeteria. And she uses an adaptive toilet in the nurse's office, something she was very adamant about. She did not want a special bathroom situation that the public could see. So we have a wonderful school who's been very accommodating for all of her needs for her to be independent in high school, which is great. Ways that we keep her active: she continues in physical therapy. Not a huge fan, but she knows it's sort of a necessary evil. She's taken private Pilates classes, which helps with her core strength as well. She rides a stationary bike at home while watching her favorite shows. She's the manager of the high school volleyball team, and that makes her feel sort of part of a team, which is great. She's on student council and joins other clubs throughout the school day. She loves cooking, doing makeup, listening to music, and watching sports. She's an avid sports watcher. And her favorite activities to be active are going to the mall and Target. So, and I'd say for anyone who is newly diagnosed with this disease or sort of starting to figure out sort of this path, I'd say the number one piece of advice I would say is connect with our community. We have met so many people from the different patient conferences that we attend. And when Sammy met some friends who have LGMD, said, I finally found people who truly get me. And that was really awesome from a parent to see that our daughter found someone that she could really talk to about her challenges. And recently at the Speak Foundation LGMD conference, she met a new friend with LGMD2A, and, zipping around and just having fun, and that instant connection was so nice. We do go to the University of Iowa Wellstone conference every year. And again, just to connect with our community is really priceless. We have a really active social media community. And so whether you're on Facebook or Instagram, you can connect with lots of different LGMD groups where we share life hacks and advice, and just sharing stories of support is really important. This is our family at the Iowa conference. I say, if you can get there, it is not easy to get to Iowa City if you have tried, but it is always worth it. We just have this amazing community that has really become like family. So we have a nonprofit organization that my husband and I started back in 2011, and we have an annual event called Connecting for a Cure. So if you want to get involved, we'd love to chat with you about that. And also, we have an amazing advocacy partnership with other groups. I can't name them all, but the ones we work most closely with are the LGMD2I Fund, the Speak Foundation, LGMD Awareness Foundation, and of course the MDA, and who was putting on great events like this today. And if you also, it's really important to get registered. We have a very important FKRP registry, and something that we support every year, and really encourage all of our patients to not only get registered, but also update their information annually. And I just want to say thank you for letting me have these few minutes today. And thanks again for your time. And I will hand it over now to Dr. Nick Johnson from VCU.
Thanks, Kelly. It's a hard act to follow. So I'm here to, actually my slides. I'm here to talk about some of the science behind, basically Sammy's story. First to say that limb-girdle muscular dystrophies as a whole represent the most common form of muscular dystrophy, and combined, the estimated prevalence is about 1 in 20,000 individuals. There's a really complicated naming system. People even use it today, talking about 2I or R9, but essentially in the new naming system, type D would be a dominant disorder. The number would give you the gene mutation, and type R would be a recessive form, and the number would give you the gene mutation. We're talking today about LGMD R9 or 2I, which is due to FKRP, represents about likely 9% of all individuals with limb-girdle muscular dystrophy. Limb-girdle muscular dystrophy, I think really Kelly said it the best, kind of describing it, but really the weakness often starts in adolescence, but it can be quite variable. It affects the shoulders and the hips first and is progressive. Somewhat unique to limb-girdle R9 compared to other forms of limb-girdle muscular dystrophy. A lot of times kids can present with what's called rhabdomyolysis, which is when people do a sport and then they have muscle pain and dark urine after exercise. And that can sometimes be a trigger for getting that genetic testing, and which is to say that the only or the best way to diagnose LGMD R9 is with genetic testing. There are now sponsored genetic testing programs that will do this for free using a saliva kit that can actually come to your home. It's super easy. If you haven't gotten, if you're thinking, well, I think I have limb-girdle, or my doctor says I have limb-girdle, but I haven't had genetic test yet, now is the time to ask about that and get that sponsored genetic test. As I said, it is a slowly progressive condition. Individuals may end up having breathing problems and may have muscle pain. LGMD R9 is caused by a loss of an enzyme called FKRP. So the way to think about it is like in old-fashioned fire brigade, you have this series of enzymes, which is shown here on the left, that are adding sugars to the end of a protein. And FKRP is one of those kind of men passing the buckets along, supposed to be adding sugars to this particular protein. And in LGMD R9, that particular enzyme is lost. It's actually part of a larger group of muscular dystrophies called the alpha dystroglycanopathies. Say that 3 times fast, because you can actually have a mutation in almost any one of these enzymes, and it results in progressive muscle weakness. The figure, what does alpha dystroglycan do? The figure on the left shows that it's, this protein right here, the kind of green circle, the bottom part there is the inside of your skeletal muscle, and the outside part is kind of what's outside your skeletal muscle. And so it's really part of a series of protein linkers you can see kind of acting as an anchor to connect that individual muscle cell to the rest of the kind of where it's sitting in the body. So that's, when you lose that protein, then when muscles expand and contract, they're susceptible to damage. They can break down over time, and that's what causes the weakness. LGMD R9, like I said, is caused, you need to have 2 pathogenic variants to have this condition, so 1 on each copy of this gene. What's unique about this particular form of limb-girdle compared to others is that it really has a very prominent, what we call the founder mutation. So there is this particular mutation called L276I, which is shown there, and that's common in Northern Europeans. And oftentimes you'll actually hear clinicians or the patients say, well, I'm homozygous, and that's really what they're talking about. Some people can have a second mutation in a different position on FKRP, and those individuals are referred to as heterozygous. That particular nomenclature really is unique, or is not unique to limb-girdle muscular dystrophy, but the way that when people talk about LGMD R9, that's what they're talking about, is whether or not you have this common pathogenic variant or not. And it turns out actually that may have some impact in how the disease progresses. These are data here from that University of Iowa study that Kelly was referencing. And you can see here across, how fast you walk, for example, or how well you can climb the stairs or walk 6 minutes. In general, you can see, for example, that there's this kind of shared line, but that people who are homozygotes, so have 2 pathogenic variants in that shared mutation, tend to have a little bit slower progression and tend to have, have a little bit more function over time. There's, this is, as you can see in the lines, this is a generality. This is in no ways, reflects an individual's particular state and where they are in their disease course. But, this is one of the things when people talk to you about clinical trials or thinking about clinical trials, this is one of the things that investigators will often take into consideration. So what do we do now in clinic? So we do have a multidisciplinary clinic. We monitor pulmonary function. Actually, like I said, Kelly said better than I could. We talk about adaptive equipment or surgeries as we need, manage the pain as it develops. But unfortunately, in 2023, I can't write a prescription for disease-modifying therapy, which I know we're all pretty excited to move forward with. However, I am excited about this technology called gene replacement therapy, which is basically where you take a virus, you take out the bad viral DNA, and you stick the gene that's missing, like FKRP, for example, into that virus, and then that allows the virus to deliver that particular gene into the skeletal muscles. Why do we use viruses? Well, as many of you who've had a cold know, they're really good at getting into your cells. They get all throughout your body. So out of all of the delivery tools that we have to deliver a gene or something else, viruses actually work really pretty well. But these are probably one-time therapies, at least they are in 2023. So it's important to keep that in mind because of the amount of virus that's given, there's a risk of an immune reaction if you were to have it receive it again. If we drill down into that, what is part of that AAV vector? This is actually what's inside that. So this is a viral capsid, and you can see that the way these therapies are done, you have something that kind of turns on the gene like FKRP here, and then there's these other pieces that complete it. So this is the entirety of what goes into a gene therapy product. There's a certain size component, which is why FKRP is amenable to gene replacement therapy, but some forms of limb-girdle muscular dystrophy are not. And at least in animal models, at a minimum, it works. These are data here from Atrium Health showing in a mouse model that received an FKRP gene therapy. You can see this panel C is the one I like in particular. You can see that you don't see a lot of staining in these mouse models, but then after you deliver it, you can see that there's much more protein expression, and that's persistent. And then more importantly, what actually happens, you can see that CK, which is a level of muscle injury, gets better after gene therapy delivery, and that the amount of time or distance somebody can run, you can see here, goes up again after delivering the gene therapy. So this is great for these mice that they were able to have this disease that was improved by gene therapy. But I think many of us are excited to see what happens in human clinical trials like the one that AskBio is conducting, because like I said, I'm very excited about the promise of gene therapy like the one for FKRP, moving beyond mice and into human clinical trials. I think beyond that, we should all be heartened by the amount of drug development in the last 2 years and the progress that's occurred there. We do need to have some patience. These early phase clinical trials, like I said, similar to the one that Ask is doing, they do enroll patients slowly. So a little bit of patience for all of us is important, because I know we're all ready to kind of see how these work. And many of us in the Grasswell LGMD Consortium have been working to develop these partnerships between individuals living with limb-girdle muscular dystrophy, researchers, and pharmaceutical companies to advance these therapies for you or your loved ones or anyone living with limb-girdle muscular dystrophy. And with that, I will pass it back to Ina.
Thank you, Kelly, as well as Dr. Johnson, for providing us with insight related to what your daughter goes through on a day-to-day basis. We know that this disease is heterogeneous, but there are very common symptoms, and really great to hear the story. And Dr. Johnson also walking us through gene therapy, and the promise of what's here to come. We do know that there's a number of clinical trials ongoing right now, not only with AskBio, but with another couple of other companies. So really a lot of work and significant contributions, and we're really excited about what will be coming in this particular disease space in the next few years. And with that, Gina, I don't know if there's any questions that have come in.
I have not seen any questions come in. If there is any specific questions that anybody on the line has, feel free to go ahead and type it in your chat. We will give it just a minute.
And this is Ang, and thank you very much for both presentations, very informative. While we're waiting for questions to come in, so Dr. Johnson and Ms. Brazo, any sort of comments or perspectives on, the issues around the diagnosis and the process of diagnosis, and that was it arduous, and any recommendations around finding a specialist and getting the appropriate care that you need?
Well, we've come a long way, and Dr. Johnson mentioned that in his talk. When Sammy was diagnosed back in 2010, our insurance wouldn't cover it, and there was no free genetic testing at the time that we knew of. So we were going to have to pay $10,000, I think, out of pocket to get her tested. But luckily, we got her enrolled in her first natural history study through the NIH, and they did cover that testing for us with Dr. Bonneman's team. So now it's so much better. Like Dr. Johnson said, you can get free genetic testing, I think through the Lantern Project still. And it's basically a kit that can come to your house. You can spit in the tube and send it in. So it's come a long way. I know other people that some patients who had questions for years did the 23andMe testing, and they've narrowed it down at least to know what type of panel to come to get tested. So there's a lot of patient stories I could really tell you all day, but I think it's come a long way. And I really encourage for those that have sort of that LGMD, and they stop there. There's a lot of patients say, "Oh, I have LGMD." Well, what type? I'm not sure. That years ago they were told LGMD, but we don't know what subtype. That I would encourage, get your subtype tested, because when you get into your micro group, first of all, the stories are so similar, and they're not all the same. Although we are an umbrella term of LGMDs, they are all similar but yet different in their own way, and the treatments are going to be different. And Dr. Johnson, the expert, can speak more to that, but that would be my soapbox.
I couldn't have said it any better. I agree.
Very cool. And Gina and Terry, did any questions come in, or?
There is 2 questions in the box. The first one is are you ready to enroll European patients in the clinical trial in the US?
And Ina, do you want to take that, or?
Sure. So we have a number of sites here in the US, and what we do is we recommend that the patients actually contact the clinical sites. There is, a number of visits that are required. So really, I think they can explain the inclusion-exclusion criteria, as well as the logistics of, the ability for you to stay at the site. So currently the trial is ongoing in the US. However, it's best that you contact the clinical sites to get particular information related to the logistical considerations.
And Ina, thank you very much. And maybe Kara can also add some color. AskBio, as part of the clinical program, the clinical trial, does provide a concierge service. We actually have had interest from patients overseas. We're still in the process of trying to get the logistics to figure out the workflows. But for example, we are open to having patients from Western Europe, Northern Europe, and Canada. Again, this is a process and evolution. And I think, first and foremost is to contact, they basically AskBio as well as our clinical investigators as part of trying to initiate that process. Maybe Kara, do you have any comments?
Yeah, I was just going to add our email address that we can provide more information to you about the clinical trials, and that is askfirst@askbio.com. So it's AskFirst@askbio.com.
Excellent. We'll have time for one more question here. Is there an age limit on gene therapy?
So I'll answer that one. In the current AskBio trial, it is adults. However, we do know that, as we continue with the program, we hope to expand on the population. And I believe it's the same for some of the other clinical trials that we're aware of. Currently, it's ongoing in adults, but I'm sure they're all also looking to expand that patient population and the age.
And thank you, Ian. And maybe Dr. Johns can also add some color, but specifically for the AskBio trial, the cutoff age is age 65 years. And the cutoff was, the rationale was around making sure there was enough potentially viable muscle for successful transduction. That is a work in progress just to figure that out. Again, this is just part of clinical trials, right, to understand that. We also understand that time is muscle. And as we learn more about the therapy and how it can impact lives, we do believe that there are opportunities to further explore the use of genetic medicines, such as for the treatment of limb-girdle, for treatment, for example, other manifestations of limb-girdle beyond loss of ambulation, for example, pulmonary function or cardiac function. Maybe Dr. Johnson, any comment?
Yeah, I mean, I think it started with this particular age group, but I expect as the safety data continues to be positive, then I think, I expect it to continue to roll out. Also, to make a comment to say that, this type of gene therapy should work for both heterozygous and homozygous FKRP, but gene therapies are very specific to the gene, so they would only work for that subtype. They wouldn't go to a different one of the alpha dystroglycanopathies, for example. That would be a new gene therapy.
Thank you. Well said.
Excellent. Well, thank you for joining us today for the Industry Update webinar presented by AskBio. We hope that you found this informative, and we hope that you join us for future education events. Thank you. Have a great day.